Finally, we're going to touch on the last two types of tissues, muscle and nervous tissue. For our purposes, we're just going to gloss over these kind of a lot. They're going to become more relevant later on in the semester, and we're going to harp on these types quite extensively. So there's three main types of muscle: skeletal, cardiac, and smooth. Skeletal muscle is what you really just think of as muscles. It's your biceps, it's your triceps, it's your abs, it's your hamstrings. These are the muscle fibers that are long and move your skeleton around, hence skeletal muscle. Major features of skeletal muscle include that they are long, straight, and parallel to one another. They have tons of mitochondria. They have tons of nuclei per muscle cell, and the nuclei are all kind of pushed off to the side of the cells, so they end up looking like flat, skinny little beads around the edges of these barrel-shaped red cells. And they're striated, which means that, like, if the cell goes this way, they've got stripes that go this way. They're striated, they're stripy, and they're kind of subtle little stripes. Which one of those three circles do you think then would be skeletal muscle tissue? Long, straight, parallel, barrel-shaped cells with perpendicular stripes and lots of nuclei pushed off to the sides of the cells. Down here at the bottom right, skeletal. Remember, for the purposes of the exam and the practical, you're going to have to give the type of muscle cell as well as muscle. You can't just say skeletal cell, and you can't just say muscle cell. You have to say skeletal muscle cell, cardiac muscle cell, smooth muscle cell, okay? Cardiac muscle tissue looks a lot like skeletal muscle tissue in that you have long sort of cells that all go kind of more or less the same direction. They have a bunch of nuclei per cell. Or sorry, they only have one nucleus per cell, but there's a lot of cells packed into the slide, so it can look like there's more. They're also, but they do also branch. So there's long cells that go more or less in the same direction, but they branch. They have one nucleus per cell, but since you have the branching, it can look like there's multiple. And they are striated. Which one of the remaining two do you think is cardiac? Cardiac muscle tissue. This one right here. You can see you have these cells that go like that. This one branches over here. And this one you can see kind of, whoops, branching off behind the other guy like that. Now you'll also notice, let me erase that so you can see it better, that you have striations that go this way. It's hard to tell on the slide, but you do. And then you have these kind of sporadic, darkish lines. This is another hallmark of cardiac muscle tissue, and they're called intercalated discs. You need to know this for the exam. So an intercalated disc is darker because it picks up dye a little bit more darkly than anything else. The striations are because fibers pick up dye really nicely, and in skeletal and cardiac muscle, you have all of your fibers arranged in the exact same direction, and they overlap at regular repeating intervals, and so some part of it looks darker than others, some part of it looks thicker than others. Intercalated discs are really the junction between one cardiac cell and the next. So if you have one cardiac cell like this, you have edges that look like that. Here's one big fat, and they're usually round nucleus, like that. The next cardiac muscle cell will join it right there at the intercalated disc. When your heart cells beat and they send an electrical signal, which is really just a big ion shift, a lot of ions moving, they're sending it along in the directions of their intercalated discs where they make those junctions. They're not sending anything like through here. That's just regular old plasma membrane. They're sending it through the parts of their cells that are the intercalated discs. If your cardiac muscle cells are sending ions through the intercalated discs, the boundary between two cardiac cells at that special spot. What type of junction do you think is holding those two cardiac muscle cells together at the intercalated disc? Gap junctions. Gap junctions. Remember, gap junctions hold two cells very closely together. They're basically like tight junctions, but they're arranged in these circles that leaves a hole in the middle for stuff to pass through. If you don't have this hole between the two cells at the intercalated discs, then things can't really move super easily through the intercalated disc. And so when ions have to move to enable this sort of electrical signal to go down the length of the heart, that's how they get through from one cell to the next, is those gap junctions. Now, when that moves and you have this electrical shock through a cardiac cell, through a heart cell, it's going to contract, it's going to shorten. So you've also got, like, you've got a cardiac muscle cell that's going to be like here, right? Kind of tucked behind those guys, and it'll have its own intercalated disc with something else. These cells are going to be connected because they're touching each other, they're next to each other, but they're not going to send signals past to one another. They're not going to move any ions between each other. You can tell because there's no intercalated disc. But they are going to both contract, and so they're going to move, and you don't want to tear the tissue. So what type of junction would you find between two cardiac muscle cells that don't share an intercalated disc? A desmosome. A desmosome. Remember those, you've got big old plaque on the inside of one cell and a big old plaque on the inside of the other cell. And they both just sort of release a bunch of fibers that sort of stick together and get tangled. And each cell can shift past one another a little bit because these have some give, but they don't slip away from each other. So you've got these two types of cells abundantly in cardiac muscle, or these types of junctions abundantly in cardiac muscle tissue. You would find, say, an intercalated, or these are your intercalated discs, so you find gap junctions there, each one of those dark, dark, dark lines. And you would find desmosomes like right here. Let's say you can see one tucking back behind another cell, maybe over here, there. You see cells touching each other, but you don't see an intercalated disc. If you're not sure if the cell is striated or not, but you see this branchiness, and especially if you see those intercalated discs, those sort of widely spaced dark lines, you know it's cardiac muscle tissue. If you can't find those lines and you're not sure if it's branchy or not, and you're not really sure otherwise, if you can't see those intercalated discs, it's not cardiac muscle tissue. If you look at an example of cardiac muscle tissue, it will always show you an intercalated disc. I've never found one that didn't. Finally, last one we're left with is smooth muscle tissue, and it's just called smooth muscle tissue because voila, under a microscope it looks super smooth. In cardiac and skeletal muscle tissue, there's striations because inside of those cells you have those fibers. And remember, fibers pick up dye really nicely that overlap in regular, evenly spaced things. So when you dye this cell, in here ends up looking really dark because you have a lot of fibers, and in here looks lighter because you have less fibers holding on to dye. So you have the stripiness. In smooth muscle tissue, your cells aren't barrel shaped, they're sort of spindle shaped. They've got these long sort of ovally nuclei, and the fibers inside of them are sort of more like crosshatchy. And so when you stain smooth muscle fibers to look at under a microscope, there's not this regular pattern that creates any kind of stripiness. So the dye takes pretty evenly and it looks very smooth under a microscope. These cells are spindly, as I mentioned. There's no striations. They don't branch, but they do get squashed up against each other. So you can kind of see like there's a few different, like they're all generally going this way, but they kind of undulate past each other a little bit. Their nucleus is in the center and it tends to be sort of ovally as well. You find skeletal muscle tissue, as we mentioned, attached to bones. Cardiac, you only find in the heart. In the heart. The heart itself is cardiac muscle tissue. Your major blood vessels, your aorta, your vena cava, your other arteries and veins and capillaries do not have cardiac muscle tissue. It's just the heart itself. Your blood vessels and your gastrointestinal system and your urinary system and all those places that are involuntarily moving. Like you can't just will your blood vessels to dilate, right? You can't will your digestive system into moving faster or slower. It just will if it's going to. All those places where you have movement in your body that you can't control are smooth muscle tissue. And finally, we have nervous tissue. Two major things that you need to be able to identify with nervous tissue: a neuron and neuroglia. I'm not going to make you identify individual neuroglia or be able to tell me anything about different types of neuroglia for an exam, for the lecture exam, okay? You will for the neuro, so I'm going to introduce the names of some of these, but you don't need to memorize them until later. The neuron itself, though, is the thing that's actually going to transmit electrical impulses and neurotransmitters and stuff to send signals around your body. Each neuron has a classic star-shaped cell body that receives a signal. So you've got these long wispy guys coming off of the main cell body. Here's your nucleus and your nucleolus. And those are called dendrites. And then there's a long tail that comes off of that cell body, and that's called an axon. And basically what this arrangement is you have, through the dendrites, input signal, consolidation of signal through there, and then sending of the signal through the axon. If I were to draw out the whole thing, you usually only get part of a neuron for any given slide, but it'll usually look something like this, where you have this star shape, there's the nucleus, and then a long tail, and then at the end there's this branchiness to it, so that one neuron can get an input a bunch of different places and give an output to a bunch of different other neurons. So each neuron is super, super well connected with a bunch of other things around it. Interestingly, though, neurons don't directly touch other neurons. You kind of talk about them as if they do, but in reality they don't. There's a little space between the end of one neuron and the beginning of the next neuron, and that space, that gap, is called a synapse. Neurons are finicky, sensitive structures. They're finicky and sensitive cells, and they need special helpers to be able to do their task. They're very, very highly specialized. And there's a host of other cell types that help support the functions of the neuron. Those are called neuroglia. You'll sometimes see that written as just glia for short. For the exam, you have to write out the whole thing. Different types of glia can do different things. So some of them make myelin. Myelin is like an insulating sheath that you find down the length of the axon, so that when you whip the signal down that whole long length, which can be like 4 feet long, depending on what neuron we're talking about, that individual axon can be like super, super long. You don't want to have signal loss down that, right? And you want to make sure that signal moves fast. You prevent signal loss and make sure your signals move fast in the electrical cables in your house by insulating them. And you do the same thing in your neurons. And that's with myelin. So some of them make myelin, some of them make cerebrospinal fluid, which is that fluid bath that your brain uses to move nutrients and wastes around inside of itself, so it doesn't have to talk directly with the bloodstream, which might have germs and stuff in it. It has its own special immune cells, like microglia, that come and go and help break down old dying neurons and break down damaged myelin and help get rid of pathogens that might have gotten in. And you have other cells that can serve as the blood-brain barrier, that can act as a connector between the blood and the neurons so that it can filter out and make sure that bad stays out and good comes in. Those are astrocytes. Your microglia right here are your immune cells. You don't really have anything else that are shown on this slide, but a bunch of different types of glia. There's not just one type of glia. This concludes all 4 of our major tissue types. And again, for the purposes of the regular lecture exam, the multiple choice and short answer question combo situation, you're going to need to know mostly just the functions and roles and general relationship between the tissues. Like, what does it do and why does it do it? What's in it? For the lab, it's going to be very strongly focused on visual identification of tissues and identification of patterns of tissues. So learning what is often found next to what and why.